This is CGTN, China Global Television Network.
I'm Emma Keeling, trying to keep up with the new coronavirus variants to find out if the vaccines will still be effective.
And I'm Jo Colan in the city of Liverpool, where you may be surprised to know they're working on a universal anti-venom that could save hundreds of thousands of lives.
The coronavirus family contains many viruses that can be broken down into smaller groups called species. One such group is the severe acute respiratory syndrome-related coronaviruses, or SARS-CoVs. And within this species are two notable strains, the original SARS-CoV from 2003 and the more recent SARS-CoV-2. Each strain can have multiple variants. Variants emerge during virus replication through small changes in the genome called mutations.
We have seen many variants over the course of this pandemic, and that's because viruses mutate, and SARS-CoV-2 mutates regularly. So we frequently see new variants emerging. It's just that most of these variants don't change how the virus behaves. And so there's no real selective advantage and no reason why these variants would propagate instead of any other variant. So we don't really pay a lot of attention to them, except for doing surveillance to make sure that we're not getting dangerous new mutations arising.
But in the last 6 months, mutations have caused several worrying new variants to emerge around the world. Most recently, B1128 in Brazil, for which very little is still known. More studied are the two variants B117 identified in the UK and B1351 in South Africa. Scientists are worried about how rapidly these variants are spreading, or what's known as their increased transmissibility.
Basically, anything that affects how the virus replicates, how the virus is released from cells, and how effectively virus that's, say, in the air can get into a new host and start a new infection. Anything that increases how effectively the virus does that can increase transmissibility.
One previous SARS-CoV-2 variant, D614G, has been found to be able to bind to human cells and enter them more successfully, suggesting increased transmissibility. It became the globally dominant strain in 2020. However, one recent study analysing over 46,000 genomes of SARS-CoV-2 found that the dominance of D614G was more likely due to what's known as the founder effect, where the variant emerged early in the pandemic and grew in frequency as the virus spread around the world. Variants can also get lucky, emerging alongside behaviour changes such as people going on holiday again, no longer wearing masks, or socialising more readily. What do we know about the transmissibility of the B117 UK variant so far?
The variant seems to transmit much more effectively, between 50 to 70% more efficiently than we've seen with previous variants. That is correlated based on what we've seen in terms of the epidemiology of the spread of this variant versus other variants within the UK and specifically the Southeast. And it looks like that variant is more transmissible, but we're really waiting on laboratory experiments to confirm that.
What do we know about the B1351 South African variant so far?
We know a lot less about the South African variant than we do about the UK variant. Now, we know that the variant has mutations also in the spike protein and in this receptor binding domain. So there's a very good likelihood that this variant is in fact also more transmissible, and more transmissible because it binds better to this ACE2 molecule on human cells. But we don't know much more outside of that.
ACE2 is a protein on the surface of human cells. It's found in our lungs, blood vessels, kidneys, liver, and even heart. The spike protein on the surface of SARS-CoV-2 binds to the ACE2 receptor like a lock and key. This is how the virus enters the human body. Genomic analysis of the B117 and B1351 variants can help provide clues about transmissibility, vaccine evasion, and disease severity through examining their mutations. In the case of B117, 17 of those mutations occurred all at once, an unprecedented amount in the COVID-19 pandemic to date. Most interest lies in mutations that occur within the genetic code for the spike protein, which the virus uses to enter human cells. Both variants have several changes in this region. One identical mutation they both share is at the 501st link in the spike's amino acid chain, called the N501Y mutation. This sits within an area called the receptor binding domain, where the spike binds to the protein ACE2 on human cells. The naming N501Y indicates that the amino acid N, known as asparagine, is being replaced by Y, known as tyrosine. Ravi Gupta, a professor of clinical microbiology at Cambridge University, has been investigating these mutations in both variants.
501Y was a mutation that appears to increase the binding between the virus particle and the ACE2 receptor on the surface of cells. That's been demonstrated. It also was associated with more severe infection when in a mouse model, but then again, mice are difficult to infect, and so it just helped the mice get infected.
Along with N501Y, the South African variant B1351 has two further changes in this region, K417N and E484K.
This is a trio of mutations that are in the receptor binding domain, which is a key target for our vaccine responses. And there are a number of studies that show that 484 in particular can lead to a degree of immune escape from serum from recovered individuals.
It's a worrying indication that E484K may help the virus evade the body's immune response and weaken the effectiveness of vaccines. B117 does not share these mutations, but has other changes elsewhere in its spike protein. P681H, where amino acid histamine replaces proline, occurs on the edge of the region coding for the furin cleavage site. This cleavage site is unique to SARS-CoV-2 and is potentially one reason why it's more transmissible than the original SARS. It also has a deletion of 2 amino acids at 69 and 70. This has been found in other variants throughout the pandemic and often occurs alongside other significant mutations. This raises the question of whether it could be a combination of mutations that causes increased transmissibility in variants. So how did B117 gain so many mutations all at once? One theory comes from a paper exploring the treatment of chronic COVID-19 in immunosuppressed patients using convalescent plasma. This involves taking plasma from patients who have recovered from COVID-19 and giving it to those suffering with the disease to try and provide a form of artificial immunity.
And that is perfect for the virus to then just keep going and then acquire mutations. So I think the biggest problem is chronic infection as a source for these new variants. Chronic infection is most likely in the context of immune suppression, and we potentially could exacerbate those issues by giving people with terrible immune systems therapies such as convalescent plasma. But on the other hand, there is a clinical need and people perceive that they need to be doing something to treat these individuals. And so we need to be doing such, if we're going to do this, we need to do it in a controlled environment where we minimize the risk of passing on those variants that are generated.
The use of convalescent plasma as a treatment for COVID-19 has come under renewed scrutiny after researchers in the UK concluded a major study, finding that its use had no significant difference on patient outcomes. But it's not just convalescent plasma giving birth to these new dangerous variants. As SARS-CoV-2 continues to reach ever higher global case numbers, the power of virus evolution will continue to more rapidly create mutations that could change the course of the pandemic. This can be seen with the new variant B1128 in Brazil.
This has some concerns about it because it has the 501 and 484 mutation. And then the third mutation it has is position 417, which is also mutated, but a little bit differently in the South African strain. So there's this different amino acid replacement, again suggesting that these are common mutations that the virus can pick on to do the thing that it needs to do, which is to escape immunity.
So what can we do?
We do need to step up the vaccine programs, as fast as possible. And I think any vaccine, even if 50% protective, will be better than nothing. But we need to be following this up with better vaccines that cover these mutants that are targeting other parts of the virus as well. It's the next sort of phase of vaccination.
With the initial hard work already done, one vaccine company, BioNTech, say they can update their vaccine in just 6 weeks to respond to any new variant that reduces efficacy. In the future, this could become more like traditional flu vaccinations, which are updated each year. But with this SARS-CoV-2 virus currently running rampant around the world, is there any way to prevent new vaccine-resistant variants evolving?
There's no way to prevent mutations. Mutations are just going to occur. What is important to do in that context is that we make sure that we are continually doing surveillance and sequencing the virus to ensure that we're really aware of any mutations that are coming to light. Now, it's important to note that most of the same precautions that we already have will still be effective. This doesn't change how the virus spreads. So wearing masks are still effective. Keeping distance from others, avoiding crowded indoor spaces, all the things that we've heard about for many months now, these are still effective.
You can see more of Razor on our YouTube channel. Search for Razor Science Show and it will take you straight there. Remember to subscribe and hit the bell button for notifications.
The World Health Organization call it one of the world's priority neglected tropical diseases. Up to 138,000 people die every year from snake bites due to a drastic global shortage of antivenoms. I'm Jo Colan in Liverpool, maybe not the place you'd expect scientists to be working on a universal snakebite cure, but they hope their research will help 3 million people every year. Anti-venom hasn't changed much since the early 1900s. Even the best only protect against a small number of the 250 different venomous snakes.
We're trying to develop a completely new treatment for snakebite, effective against all of the snakes in sub-Saharan Africa and India. In sub-Saharan Africa, 32,000 people are dying from snakebite. In India, 46,000 people are dying from snakebite. That's half the number of people that are dying from HIV. And people are dying in Southeast Asia, they're dying in Latin America. Globally, there's 138,000 people dying from snakebite that we know of.
To make antivenom, you need venom. And here at the Liverpool School of Tropical Medicine, over 100 of the most venomous snakes on the planet are helping save lives. So I'm about to be only feet away from one of the world's deadliest snakes, which will be out of its box and writhing around free on the floor until our colleagues here secure it. So it is a little bit nerve-wracking, I have to say.
All right. Yeah.
Antivenoms are usually only effective against bites from the species of snake providing the venom.
Right. Yeah. Might just try it like this. Got that. Okay. Just relax slightly. There we go. In and out again and in. Okay. Ready?
Paul, I know it's been rare, but you have been bitten, is that right?
It is, yes. I have been bitten by snakes in the past. The last bite was some 17 years ago from an 8-week-old baby Western diamondback rattlesnake. I had 26 ampules of anti-venom, which is quite a large amount of anti-venom. I spent several days in hospital with that, and the pain level was beyond anything I've experienced before. I felt like somebody was sticking my arm into boiling oil and grinding my bones together.
Currently, anti-venom is made by extracting venom from a snake. A horse is then injected with very tiny doses of venom that does not cause harm to the horse. This stimulates the horse's immune response to make antibodies to the venom. These antibodies are then harvested from the horse's blood. In order to get a high enough concentration of antibodies, horses must be injected several times a year over several years. It's a long and expensive process, not easily available in the parts of the world that need it most.
Africa is in an anti-venom crisis and has been for the last 20 years. And the reason is that the two best anti-venoms that were available in Africa for most of the 70s and 80s and 90s have disappeared because they were expensive. The demand for those by governments and so on was really low. And those companies, one in Germany and one in France, simply could no longer afford to continue producing these loss makers. And so they stopped production. And what happened then was anti-venoms that were less effective were coming in from other parts of the world and being used. And here now we have a problem because the supply of effective problem has stopped, replaced by other products which are some dangerously ineffective. But the doctors who are giving the antivenoms don't know that. How can they know that? And so they will give one vial of an antivenom, and if the patient doesn't recover, they will give it more vials, more vials. And
Of
so
a product that's ineffective?
Yes, and that it's causing these adverse effects.
Everything changed for Professor Rob Harrison when he got a call from the International AIDS Vaccine Initiative in San Diego.
The telephone call was, do you want to collaborate with us? And my immediate reaction...
Not on HIV, but on snakebite.
Well, exactly, that was my reaction. Why HIV? And he explained very quickly that they had technical platforms that they had developed for their HIV vaccine research that he thought we could use very usefully for snakebite.
Up until now, treatment has been based on the venom, not on the symptoms it causes, which include paralysis, asphyxiation, and unstoppable bleeding, and bleeding in the brain.
So the people who've been working on AIDS have understood more about the molecules and more about the antibodies and how they can work to interact broadly with HIV viruses, despite them changing a lot over time.
The HIV researchers found that some humans develop broadly neutralizing antibodies that prevent infection by the majority of HIV strains. Remarkably, these antibodies have found parts of the virus protein that are common among the many different strains. Such antibodies are therefore capable of inhibiting infection by any virus that has that region in common. In some cases, this translates to almost 90% inhibition of diverse HIV strains by a single antibody.
What we're hoping to be able to do is to engineer antibodies so that they can broadly recognise lots of these different toxins, no matter which snake bites a person, and so they can be neutralised in a generic manner.
It's hoped the new universal antivenom will require lower doses and will not produce the adverse reactions that antibodies from animals can cause.
Certainly we wouldn't need to take venom and inject it into an animal in the way that snakebite therapies are made today. We'd be able to grow cells that produce the antibodies in the laboratory and remove the animals from the process.
Developing a universal anti-venom has been made possible due to almost $12 million from the UK Department for International Development. Now, the World Health Organization identified 2030 as the date by which they'd like to halve snakebite deaths around the world. How optimistic can we be?
I think if there is more investment like the Wellcome Trust, if there are more groups getting involved, delivering the many different things that the WHO strategy involves, then we have a chance. But I do think it's ambitious. But what I don't doubt is that there are groups around the world who have that commitment and who will try their very best to deliver.
For more science, behind-the-scenes insights, groundbreaking research and even some fun, check out our Razor podcast. Search Razor Sounds on all major streaming platforms and remember to subscribe so you don't miss out on any episodes.
Search CGTN Europe wherever you get your podcasts to subscribe to The Agenda podcast. The Agenda with me, Stephen Cole, always gets to the heart of the story. Just subscribe today and listen now.
The collective behaviour of animals has long interested and mystified scientists, but recent developments in both AI and robotics have shed new light on the subject, allowing for a fresh look at swarm intelligence.
One of the amazing things being a scientist in our time right now is that there's this explosion of new technologies that we can use to understand how the world works.
This is Professor Ian Cousin. He heads up the Department of Collective Behaviour at the University of Constance. They use advanced technology to investigate patterns that are formed in nature.
Because we're interested in principles of collective behaviour, we actually study an extremely broad range of organisms. So we literally study the simplest animal on the planet. It's called Placozoa. We study fish schools, bird flocks, all the way up to human crowds, because we ourselves, of course, live in a collective. And so across these different scales of biological organization, we can use the same types of technologies, the same sort of tracking and imaging tools to really sort of understand how collectives operate.
Most recently, Professor Cousin's team has been investigating why fish school.
If you're already in a school, if, for example, the threat of predation forces you to move together, then there may be other benefits, such as exploiting the hydrodynamics, the way that the water flows around other individuals.
Just as dolphins swim alongside boats or cycling teams form a peloton, it's possible that fish benefit energetically from schooling.
As a fish swims, it swirls the water behind it in what are called vortices. And then there's the potential for an individual following this fish to exploit the energy from those vortices to save energy as it moves.
Testing this notion has proved tricky, so the team has created a robot to help.
We cannot sort of ask a fish, are you saving energy or not? And what we can do, however, is we can develop what's called a biomimetic robot, a robot that swims like a real fish. It really behaves like a real fish. And the great thing about the robot is we can measure precisely how much energy it's using. So by building multiple robots, we can create a school where we can measure the properties that in the real world are very hard to do so.
The team experimented by putting pairs of robotic fish together, varying their swimming style and recording the data, which revealed something wholly unexpected.
We know that when fish swim, they're very dynamic. They're in all sorts of different positions with respect to each other. Previously, it was thought they have to be in a very fixed position if they're going to get hydrodynamic benefits. And what we showed was, if the fish are side by side, then synchronizing is the best thing to do. But as the fish falls further back, then actually anti-synchronizing is the best thing to do. And in between, there's an intermediate. And so just by adjusting the way the follower beats its tail with respect to a leader as a function of the distance from that leader could allow the fish, irrespective of where it is, to always be able to save energy.
The scientists were then able to observe this underlying rule in the behaviour of real fish.
It was wonderful because the real fish were following this rule extremely nicely. We had this new way that it should work, and indeed nature has found that solution.
How have you managed to compare the robotic data with data from observations? Have you had to use more technology in order to correlate everything?
We needed to use computer vision, a computer to sort of track the motion of the fish and also to get the body posture. How are they beating their tails? And so this gave us the information we needed to test our ideas with the real animals.
Professor Cousins' team are using deep learning, which utilises artificial neural networks to solve complex problems.
Relatively new technology over the last 5 years or so. I mean, this is how our phones know how to recognize our voices and so on. And we use this to actually track the animals and understand how they're behaving. And it's really revolutionized the study of animal behavior.
AI and robotics are not the only technologies that the team uses. A 3x3 metre digital aquarium also allows researchers to examine the way fish respond to visual input.
Using our multi-point projection system, we can project videos onto the bottom of the tank to influence the behavior of large schools of fish and ask how they can process information. Here, for example, we've induced the swarm to spin, and by reversing the direction of rotation of the dots, we can induce a directional change in the group.
As well as creating the world's most high-tech fish disco, Professor Cousins' researchers have been pushing the boundaries on interactive holograms.
One technology we've invented is immersive holographic virtual reality. So I had this dream that, what if we could create photorealistic holographic animals? So the animals are interacting with holograms in real time.
How this works is that basically they're in two different bowls, they're in two different environments, but they're in the same virtual environment. So in order to create an illusion of a 3D object, of an object that's popping out of the screen and it appears to be in the same body of water as the real fish, the projection needs to change in real time according to the position, according to the perspective of that one individual.
Basically, Professor Cousin and his team are developing technology that takes the scientists out of the equation, so the group behaviour of the animals can be monitored without concern for how it might be affected by an observing scientist.
If I'm influencing you and you're influencing me, it's kind of hard to work out who's sort of really, in control, who's influencing who more. If there's a third person, am I influencing you via that third person? Or am I, it becomes very difficult.
The technology is absolutely amazing, but living in the commercial world, the big question is why?
People seem to think that, there needs to be a reason, a direct benefit to humanity for research. But in actual fact, if we look at some of the major innovations in science, for example, the structure of hemoglobin, that was studied because it was interesting, not because someone thought there was a direct benefit. The structure of DNA, for example, was studied because it was known to be important for biological life on the planet. And so one of the very important things about science is to give people freedom to explore and to study very broadly how the world works, rather than focus in on it's got to relate to us now. I've always been driven by a fascination of nature rather than direct application, and both are important, but they can work in synergy. So our work has had unanticipated benefits, both in terms of, say, robotics, but also in terms of algorithms in computers that can then search for data, and in actual fact having these algorithms function as swarms. But that wasn't the reason we found those algorithms. We found them because nature had found this amazing solution to a challenging problem, and then we could utilize that.
And Professor Cousins sees very specific applications for his school of robot fish.
Our work is able to very seamlessly translate to collective robotics, how we can create swarms of robots that can use the sort of collective intelligence of animal groups to help us understand the world, to help us explore dangerous places, and to get information about, for example, how humans are changing the climate and the oceans. And so there's lots of benefits for this, but I think the most important thing is just that it's interesting.